Dude, so the next important the next important macromolecule that you have to know is carbohydrates. Um, a lot of times we think of carbohydrates as the thing that provide us with energy, but that's not the only thing that they do. And we have to learn how to classify them so you can just look at them and be able to tell that that molecule is in fact a carbohydrate. So let's go over the rules. How to identify carbohydrates? The first thing, CHO, again, just like lipids, a carbon to hydrogen oxygen, but in a 1 to 2 to 1 ratio this time. So there's equal amounts of oxygen and carbon. Think about the famous carbohydrate you learned freshman year, glucose. C6H12O6. Notice what the ratio of the carbon, hydrogen to oxygen is, 1 to 2 to 1. And you can test it and check it out for all of these different examples that we have presented here. Remember, again, if you look at a stick diagram, anytime you have like a point, that's a carbon, and then that's the 4 valence or the 4 bonds that comes off that specific carbon. Here's our glucose molecule, carbon, 1, 2, 3, 4, 5, 6, 6 carbons. How many hydrogens? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and then oxygens 1, 2, 3, 4, 5, 6. Okay, so it follows those rules. A lot of times, though, carbohydrates may be drawn as these ring structures. It's like a hexagon with, or a pentagon sometimes, with an oxygen in between, and then they're connected to each other. So they look like long chains of these same sugar molecules repeated over and over again. So carbohydrates, as I mentioned before, serve as fuel. Those are like glucose. It's a molecule that is used in cellular respiration to generate ATP, right? But all sugars can actually, or I should say a lot of sugars, can be used as fuel in our bodies as well. The monomer of a carbohydrate is a simple sugar. It is one individual sugar molecule. Okay, so glucose is a monosaccharide, or a simple sugar. Fructose is a monosaccharide. Galactose is a monosaccharide. It's just one individual unit in the name of that unit. So as I said here, monosaccharides, one single sugar. It is the simplest of all the sugars. It can be used for fuel directly. We can actually take that glucose molecule and convert it into other types of molecules in our body, which is really interesting. Then you can take that glucose molecule and combine it into polymers, which then we would go beyond being just a simple monosaccharide. Before we talk about those different types, I want to talk about how sugar bonds. So sugar can bond together using special covalent bonds called glycosidic bonds. There's two types, alpha and beta. Now we can break down alpha glycosidic bonds fairly easily, and those are found in things like starch and glycogen, which is in our muscles. We can take starch, like in bread, and break it down into sugars, and then use those sugars as fuel. But beta-glycosidic bonds we cannot break down as effectively. We have one enzyme that can break down one type of beta-glycosidic bond, and I'll talk about that in a second. But most we cannot break down. When glucose molecules, for example, are bonded together with beta-glycosidic bonds, it's called cellulose. Cellulose we cannot break down. It's like fiber, and that is what ends up going through our bodies and ends up in our poop. Okay, so alpha glycosidic bonds, which kind of look like this. And you don't have to memorize these bonds and the structures and what they look like. That's not what the AP Bio exam would ask you. But they would give you a scenario maybe where they talk about these different types of bonds. They'd explain them to you. And then they talk about an enzyme and like why would one be specific to the other. And we'll get into that when we get into enzymes. But remember, you don't have to memorize this. This is just for some background information. So you can see here, if we take one sugar molecule and bind it to another, in this case via a beta-glycosidic bond, we get lactose, which is a sugar that is found in milk. Remember, I said you cannot break down beta-glycosidic bonds. This is actually the only exception. We do have an enzyme that we produce in our bodies that can break down lactose, but it's supposed to be shut off after infancy, after you are weaned off of your mother, so you're no longer breastfeeding. But in European countries, what happened was, instead of getting shut off after that time, it maintains its onness. So it stays on. So we continue to make lactase to break down lactose for our entire lives. And some people it shuts off though, and they're called being lactose intolerant. Okay, and so being lactose intolerant is actually much more common. It's only in people of European descent that lactase tends to stick around and be turned on for your entire life. So here's kind of explaining that whole thing. So lactose sugar by itself, because of that beta-glycosidic bond, we don't have any other enzymes other than this lactase enzyme to break it down. If you put lactose through your body and you don't break it down, it actually irritates the intestines. And then it's broken down by bacteria, causing bloating and gas, and you get inflammation, and that's what causes the pain that you get when you have lactose intolerance. So what do people with lactose intolerance do if they like drinking milk? They take an enzyme called lactase, as we mentioned before. So if their body doesn't make it, you can actually buy it synthetically. That's what lactate is. Lactate is an enzyme that breaks this down into its component sugars. Polysaccharides are now taking more than two sugars and putting them together. So a lot of them serve as energy storage in us, like for example, starch in plants. In us, it's called glycogen. In plants, what we can also get is cellulose, and cellulose is a special type of structural carbohydrate. It's not used for storage of energy, it is used for actual structure in the cell walls of plants specifically. Remember, look what it has. It has beta-glycosidic bonds. We cannot break those down. So that, again, as I said, is the roughage or the fiber that we eat in our diets. But we can break down alpha-glycosidic bonds. The only difference between cellulose and starch which we can use for energy, and cellulose that we cannot, is the fact that there are different types of bonds, alpha in starch and beta in cellulose. So this is showing you starch specifically. You can see here's the alpha-glycosidic bonds right here. Sometimes it's called amylose. It's just another name for starch. And as I said, it is easy to break down. We can break down starch in our bodies. But we cannot break down cellulose. Glycogen, it's very similar, except we use it as a storage molecule of sugar in our bodies, in our muscles, for example. It's a little bit more branched than starch is, but it's still all alpha-glycosidic bonds, so our body can easily break it down into its component parts in our muscles when we need to use glucose for fuel. This is showing you cellulose now, a little bit different. As I said, it has beta-glycosidic bonds. Why do you think, or why can't we break it down? It's because of those beta-glycosidic bonds, as I mentioned previously. It just forms these long chains of fibers that the plants will use in their cell walls. So that being said, some animals can actually break cellulose down. Cows do that, for example, but it's not because the cows have the enzyme to break it down. It's because they have microbes, bacteria that have the ability to break that down. And what cows will actually do is they will swallow and they will chew on grass. They will then put it into one of, you've maybe heard that cows have multiple stomachs. They'll put it into one of their stomachs and they'll churn it and mix it up with bacteria. And then they'll throw it up back in their mouth and chew on it for like a half hour and swallow it and then mix it up and throw it back up until finally all of the bacteria have broken down that cellulose into the component glucose molecules. But the cow cannot do that without the help of these bacteria. So it's kind of an interesting side note there about cellulose. Chitin is probably something you've never heard of before. Chitin is actually found in the cell walls of arthropods, like insects, crabs. So like when you think about the exoskeleton on a crab or a lobster, that's made of chitin. And it has beta-glycosidic bonds in it. So it cannot be broken down by us either. But sometimes they actually use chitin to be, for surgical, what are they called? Dissolvable sutures and things like that. All right, so a quick review of carbohydrates. Carbohydrates are used for energy, and they're also structural, right? It's a CHO 1:2 to 1 ratio, and you tend to look for pentagons and hexagons connected with an oxygen in between them. They are linked through dehydration synthesis by alpha-glycosidic or beta-glycosidic bonds. Alpha-glycosidic we can break down, beta we cannot. And they're classified based on the number of sugar molecules that are there. A monosaccharide is 1 sugar. A disaccharide is 2 sugars linked. A polysaccharide is more than 2 linked together. Usually they won't do just 3. Like a trisaccharide, it's usually multiple linked together. And starch, glycogen, and chitin would be examples of those, as well as cellulose. All right, that's it on carbohydrates.